Hi everyone, I'm Carol. Today I'm gonna do a presentation on the research that I'm gonna do in the future. So the research about the correlation between Pseudomonas edibility to discoideum and the abundance of the Pseudomonas spontaneous gag mutants. To give a basic background information, the discoideum, which I'll refer to as dicty in the following talks, is a soil drilling social amoebae that preys on bacteria, which basically means they live in the soil and eat bacteria as food. Pseudomonas is a genus of bacteria that can both, like some of the species can be edible to dicty, or some of them can be inedible to dicty. And the gene I'm looking at is called GAC-K-GAC-S. It is a two-component regulatory system in Pseudomonas that controls the secondary metabolism. And this set of genes also exists in many other kind of bacteria. So it's a quite common gene in the bacteria. So what this gene controls the secondary metabolism, and that secondary metabolism can secrete secondary metabolites, which include antibiotics and many other different chemicals. So these secondary metabolites can help the bacteria, like the Pseudomonas, to resist predation by dicty. So basically, secondary metabolism is a very useful and important component of bacteria to help them compete with each other during natural selection and help them to avoid predation. So for the hypothesis, I'm gonna look at the abundance of spontaneous gag mutants and how they correlates with the Pseudomonas secretory metabolic burden and thus its ability to dicty. So this research is very significant because the predation by protists, for example dicty, if the hypothesis is true, it means that these predation can decrease the gag mutant abundance in Pseudomonas, which means that it will increase the secondary metabolites production. This can turn Pseudomonas into a better biocontrol agents, as currently many studies have shown that Pseudomonas can live with plants and they can secrete many different chemicals from their secondary metabolism that help the plant grow and help them defend other pathogenic bacteria and fungus. So it's quite important. And for the method part, I'll choose 17 wild clones of Pseudomonas. These clones are all collected from wild clones of dicty that collect directly from the soil. And then I'll do a gag mutant abundance test by using the milk agar, which can test the X-protease expression, and the X-protease expression is controlled by GAC-K-GAC-S. So that means if there's X-protease expression, which means there's a halo on the milk agar, that means this bacteria colony has wild-type GAC-K-GAC-S gene. So if there's no halos, it means it's a GAC-negative mutant. So I'll first grow the Pseudomonas on an agar plate, and I'll set two parts for the abundance test. One is grow it together with dicty, one is only the bacteria. And then I'll do a serial dilution, as you guys can see here. And that can make the colony more visible when I grow it on milk agar. And the abundance will be calculated by dividing the number of mutants by the total number of colonies on the agar. So after that, I'll do an edibility test, which has similar techniques. I'll also grow the dictee together with bacteria, and I'll put them on SM5 plates, which is just a nutrient-enriched plates. And if there's like after growing in a 30 Celsius degree incubator, if there's only bacteria grow on the plate, it means that these bacteria is not edible for dictee. If there's fruiting bodies of dictee and bacteria both present, it means these bacteria are edible, because the dictee can, by forming the fruiting bodies, it means the dictee can grow pretty well. So that means it has food, it can get energy, so it means that they are eating the bacteria. So to look at the results, basically from my inspection, the higher GAC mutants abundance on the milk agar was only Pseudomonas present, so it means there's no selection pressure, will correlates with larger decrease in GAC mutants abundance when the Pseudomonas are grown together with dictee, which means that the bacteria are under strong selection pressure, so they need the secondary metabolism, they need the GAC gene, and that also correlates with lower edibility of the bacteria, which means the bacteria are better at avoiding predation, and just vice versa. For the conclusion, the GAC and GAC system is important for Pseudomonas to survive predation by dictee, and this predation is definitely a strong selective force against GAC negative mutants.